Hello, this is Tracey Tokuhama-Espinosa, and in this week we're going to be discussing the basics of neuroanatomy. We're going to look at this through three different approaches. First, we're going to begin with just a big word of caution. We know very little about the brain, but we're going to try to share what we do know and what's up to date at this stage. To do that, we want to put this in the context of a history of brain imaging and how that's actually changed the way that we have access to different types of information and how we are now speculating about different ways that the brain is connected and how it learns. And finally, we'll be looking at neuroanatomy, neurophysiology, and some of the basic terminology that we want to be able to use with you throughout the rest of the semester. And we'll be giving a couple of examples of the neural networks that are established to date so that you get a sense of exactly where we are in history in the development of our clearer understanding or better understanding of the function of the learning brain. So to begin, we know a whole lot more about the brain now than we ever have in the history of humanity. However, we're just really scratching the surface. There's been a lot of changes, a lot of new updates about information in the brain. We've built a lot on the past. And the scary thing is, and we need to throw in this word of caution here, is that a little bit of knowledge is a dangerous thing. So if we know a teeny bit about certain brain functions, then we generalize and we can come up with maybe erroneous conclusions. So we have to be really open in the field of mind-brain health and education to adapt to new findings. So every time there is something new, we want to stay up to date with. So as Bertrand Russell says, we know very little, and yet it's astonishing that we know so much, and still more astonishing that so little knowledge can give us so much power. So we have to be really careful about the information that we manage about the brain. Use only really high quality sources to document our work, and we'll emphasize this throughout the semester with you. To add to this, there's about a century of research that's been done looking at how the brain learns, but there's very, very little information out there about how to teach towards that learning brain to optimize its potential. So we've made a lot of progress. There's a new type of movement here where we're trying to get from negative to neutral to optimizing human potential, and we hope that this is one of the big messages we can deliver in this class, that it's not just getting back to zero, but it's actually making the most of what we have in our genes, in our own potential, and also squeezing our environment for what it can contribute to those learning moments. The reason we know a lot more about the learning brain over the past few decades is basically there's a huge increase in the number of high-quality publications. But a lot of those things are dependent in turn on better technology. There's just been a huge advance in the way you can actually look at the learning brain, not only looking at how damaged brains result in deficits in human potential, but actually looking at how we can maximize potential through different types of stimulation. So to put all of this in perspective, in a historical perspective, I'm going to share a video with you, short clip with Jeff Lichtman here at Harvard University.
Trying to introduce the study of the brain to a bunch of students. I said, if understanding everything you needed to know about the brain is a mile, I asked the students, how far had we walked in this mile? I got answers, 3/4 of a mile, half a mile, someone said 1/4 of a mile. And I said, I think about 3 inches. This is unconventional science. There's just this huge gap in our knowledge about how brains work, largely because we have no idea what they actually are made up of at the finest level. When it comes to the nervous system, there are a large number of diseases where the only real sign that there's something wrong is the outward manifestation of the disease. A person is acting crazy, or they don't seem to learn very well, or their movements are disordered in some way. But if you look at their brain, with most of the techniques we have, there's nothing to see. You've got to see the wires. You just have to see them, and you have to see where they come from, where they go, what they connect with, and be able to map that out in enough detail that ultimately you will be able to render the information that goes into a brain to see the wiring diagram at the resolution of every single synaptic vesicle in every synapse. Enough resolution, we think, to see everything in a wiring diagram. I'm a lab head of a group of people who work together. It's kind of a shifting group of people, but it's difficult work and it requires many kinds of experts. So there are lots of people working together on this project.
What we're trying to understand is how cells in the brain communicate with each other. I have to look at the entire pipeline from the beginning, where we start with a mouse, make a sample, process the tissue, cut sections, and then ultimately begin imaging them. A slice is like you're cutting a loaf of bread, except you're trying to slice very thin pieces of tissue. So for a millimeter depth, you get about 33,000 sections.
Each section of the brain is next to the section that was sliced just before it, and the slice right after it is the next frame. And if you play these frames in sequence, you see the brain not over time, but over space, as if you're looking deeper and deeper into the brain. The big objects you see that appear and then slowly go out of appearance, those are nerve cells that are being cut through. And coming off of these cells are these large, light-colored objects, which are the dendrites. Every little object in here is a little wire. To keep track of these cells from one section to another, what you'd like to do is kind of color them in. So every one of these colors have no meaning other than to keep track of each object from section to section. What this allows us to do is to generate a wiring diagram based on this. We now know everything about these wires here, and from that we can generate all the connections of every cell in that area. Information forces you into this uncomfortable position where you have to kind of say, okay, I don't get it, but I know that the real world is more complicated than the way I'm thinking about it. I feel it's a very long road, and we've just started. That's my view.
So cautious optimism, okay? All right. From here, let's begin then looking at the history of brain imagery. I mean, how do we really know about the brain? And we know that even back in Da Vinci's time, they were doing sketches of the human brain in illegal autopsies that were occurring. And I want to bring us all the way up to date through what's called the Human Connectome Project, which is trying to map healthy human brains doing certain functions so that we can try to get a general sense of certain neural networks that are common to most human beings. So this is based a lot on technology, as I mentioned before. There's been a lot of changes in the way we are able to look at the brain. So if you recall in the video, there was a mention of looking at things over space. And so there are other types of imaging that looks at things over time. So different types of imaging techniques, PET scans, EEGs, fMRIs, the electron microscopy that you were looking at just now when you were looking at the actual synapses or document how different groups of neurons connect with each other at a very microscopic level. These are different types of imaging techniques that actually make studying the brain very different depending on what level you're actually accessing information. And what we'd like you to do in this course is to be familiar with a few of the imaging techniques, because different levels of analysis are going to be able to tell you different types of information. And so using the data in the right way based on the information that you have. For example, if you have a PET scan, it's really hard to talk about synapses because you're not going to be at the right level of analysis for doing that. So we'd like you to keep these different types of techniques in mind. So where does all of this come from? As we mentioned, there are some beautiful drawings that were done by Leonardo da Vinci, Christopher Wren, Thomas Willis, but we began this modern study of the brain or of the human mind with phrenology back in 1796. Franz Joseph Gall thought that by touching all the lumps and bumps on your skull, it could actually tell your personality. There were even, unfortunately, in some newspapers, ads taken out sort of trying to explain how anybody could read the character of another person just by looking at the size of their forehead or the bumps and lumps that they had in the back of their heads. And so anyways, these things were common not so long ago, around 150 years ago, we were still believing this. Now, autopsies have been around for, thousands of years. The ancient Egyptians used to actually extract the brain through the nose. I know it sounds kind of gross, but anyways. So there were experimentations with autopsy very early on. After that, there's a lot of documentation by Galen, really fascinating work showing how the mind and the body were connected. We have beautiful, beautiful paintings, if you can call it that, by Rembrandt, drawings by Christopher Wren, Thomas Willis, as I mentioned before, trying to document the human brain based on autopsies that were performed at the time. Maybe one of the most famous autopsies ever to occur was of Einstein's brain, which was actually photographed pretty good detail for the technology of the time by Thomas Harvey in 1955. And autopsies still occur today, not with a huge amount of frequency. Thanks to technology, we have other ways of looking into the brain. In the US, about 11% of deaths do include an autopsy. It's a lot lower in other parts of the world, like the UK, which is under 1% of the deaths, because they're only those that are considered suspicious deaths or whatever, so not really for research purposes. So basically autopsies still exist today, but they're not in such high use because we have other ways of looking at the brain. If you think about the basics of the brain, there's at least three big ways you can measure it. One is electrical, the other is chemical, and then the other has to do with the general physiology and the structure of the brain. And the physiology includes things like changes in white matter concentration, which we'll talk about in a second. So the electrical changes in the brain are one way to measure brain activity, or what parts of the brain are being stimulated at what moment. So EEGs have been around since 1875. They actually measure all the different electrical waves that are in your brain: delta, theta, alpha, beta, gamma, and mu. And by measuring the different electrical impulses, you can actually tell whether or not certain areas of the brain are being stimulated. It's still very popular with very young children, for example, because it's relatively non-invasive, and you can get a good sense of things that you wouldn't be able to find out with young children. For example, a child who has had meningitis, less than a year old, doesn't talk. You can't really ask the child, well, can you hear this? Can you perceive this? But you can do an EEG on this kid, and you can actually see if different sounds are stimulating different areas of the brain. So this is still used very, very frequently because electricity is one great measure of brain activity. Beginning in the 70s and 80s, there was a big boom trying to harness a new type of imaging technique, fMRIs, functional magnetic resonance imaging. If you do a quantitative look at studies in neuroscience, you'll find that a huge number of studies are done using this imaging technique, basically because it's relatively non-invasive. The person just lies in the scanner. You can get them to do all kinds of different things, multiplication tables, play music, talk, so that you can actually do scans pretty thoroughly using fMRIs. And they are pretty detailed, so they are something that's basically very, very popular due to their non-invasive nature. fMRIs basically measure movement and blood flow, so their blood oxygen level dependent contrast. So it's kind of doing a subtraction problem where you're looking at a resting state versus an activity. So you'll take a scan of somebody not doing anything, and then you'll have them do an activity, and then you can subtract away the differences between those two and actually show which areas of the brain are active when a person is conducting a particular activity. So there's a couple different ways you can look at MRIs. There's MRIs, fMRIs, there's diffusion MRIs. These are all slightly different. They do slightly different things. MRIs basically view the anatomical structure. fMRIs are looking at metabolic function. And diffusion MRI can actually detect abnormalities at the level of white matter fiber in the brain. So they all do slightly different things, but they're basically for the similar purpose of examining physical structures in the brain. So it's not measuring electricity, it's not measuring chemicals, it's actually looking at the different physical structures of different aspects or different parts of the brain. The use of MRIs were actually kicked up a scale in the late 90s when we actually had much better neuroimaging techniques where we could do 3D scans. So after getting an MRI scan, you could actually plot this into a 3D scanner so that you can actually place the physical location of different brain areas and what abnormalities might be found based on the scans. Magnetoencephalography was started in the late 60s. It was meant to step up the scale of what an EEG would actually measure. So it's measuring, again, electrical currents in the brain at very, very microscopic levels. So this is a lot more sensitive, a lot more, and perhaps a bit more precise than a general EEG. So PET scans began in the late 1950s, and they're still used today. PET scans are conducted by injecting a nuclear isotope into a person's bloodstream, and that sort of has a life of around 15-20 minutes, but basically as it travels up into the brain, and then you ask them different questions or you have them do different things to actually see where or which areas of the brain are being stimulated, basically because they light up on the screen. Of course, they're colored in afterwards so that there's a greater contrast, but basically you can actually look at different parts of the brain or brain functioning under similar conditions for different types of brains. So a normal person or somebody with a mild cognitive impairment versus somebody with Alzheimer's conducting a similar activity, reading a sentence from a book, for example, you can actually compare and see what areas of the brain might be affected due to different situations. And most of the different technologies that I'm mentioning right now actually are not only solely used for the brain. The brain is an organ, and so almost any other organ in the brain that could benefit from these types of scans or the detail that these show you can be used for other organs in the human body as well. Technology has advanced so much that we've actually gotten to the stage of commercializing brain scans, believe it or not. Functional near-infrared spectroscopy, for example, is used in neuromarketing, believe it or not. It's only measuring what's going on in your frontal lobes. It just measures blood flow. So basically, if you're doing heavy frontal lobe decision-making types of things, it just measures that. Kind of crazy, but you can buy this online if you'd like to do your own brain scans. Very, very interesting, though, that the technology to actually do brain scans has become more widely available. And part of the difficulty in doing better research had to do with costs. Very few universities could invest in the high costs of a big FMRI machine, for example. But these types of things are quite affordable, and so many people are doing studies now based on this type of technology as well. Optical tomography was developed by Hitachi in the late 1990s, early 2000s, and this is a non-invasive technique. You can actually wire up this little girl right here, and you can have her learn to play a song on a guitar. So that where there's changes in blood flow, you can actually say, well, this area of the brain is being more stimulated by this particular activity. We can really say, however, that a great deal of the advances have really just happened in the past 15, 16 years. The Human Connectome project, this idea of studying not only damaged brains, as we used to do with autopsies, for example, or looking for irregularities in the brain in fMRIs, tumors, for example, or trying to determine problems or identify what areas of the brain are actually not functioning well in Alzheimer's, as we saw in the PET scans. The Human Connectome project has a different objective, and that is to actually document hundreds, if not thousands, of normal human beings undergoing normal activities in order to see what the typical networks might be for certain activities. So many of the scans that you'll find online on their webpage have to do with research being done at Washington University, University of Minnesota, and Oxford, in which they're trying to look at healthy subject doing simple things like arithmetic so that we can look at the brain and see what types of networks then would we consider to be typical, or what areas of the brain tend to be typically connected and in what order with different types of human learning activities. So I really encourage you all to explore this page. There's a huge amount of information there. And in fact, all of the data is shared. Every time there's a new project or something new that comes up, they actually share the data, and it's available for reinterpretation by individuals. So hopefully you'll pick up on that and be motivated to look at the most up-to-date brain imaging that's occurring these days. So what does all of this tell us? We have better knowledge about the brain and learning. So what I'd like to just caution you with is every time you see one of these kind of cartoony things, think twice. Sometimes they're being used in the right way, but most of the time they're just promoting neuromyths, and we really have to get away from that. Ideas about right and left brain learners, about men and women's learning, about compartmentalized learning, about the triune brain, that there's 3 different brains, about teenage brains. There's a lot of things that are easy to put in caricature form, and they are attractive and palatable to individuals, to teachers, to Joe Average. So many of us get tempted in using this information. However, please be cautious, because it's really never as simple as these pictures can depict. As we said, changes in technology, going from just basic autopsy structures to PET scans, we're able to develop better ideas with 3-dimensional imaging, and combining that with more advanced technologies, we're able to have a far better understanding of the brain. We're able to now speculate about certain types of connections in the neural networks that can be found in the brain, and we're actually able to document, hubs, where are the basic or where are the main crossover areas for certain types of learning. Why does The brain, when it does look at when it looks at a subtraction problem, think about magnitude, then return to symbol interpretation, then return to magnitude again. So we know that there's certain areas of the brain that are used more than others for different types of learning processes. And so in the Connectome project, you're actually able to see some fascinating links and to look at neural networks and to look at synaptic connections in the brain at a much more refined level than we've had in the past. What's most likely to occur is in about 10 years, somebody's going to make a new video and laugh at these images and think that they're as Mickey Mouse as some of the caricatures that we had in past years, because technology is advancing really quickly. But to date, this is what we know about the human brain based on these types of technological advances. And these things are giving us better hints about what are myths, what are the neuromyths, what are things that are incorrect, things about left and right brain, about using only 10% of your brain. There's a lot of myths out there. So I would just I want to call your attention to a publication that came out in 2002, which tried to help countries agree upon. There were 30 countries at the time, there are 34 right now, but the 30 countries in the OECD nations agreed upon a certain terminology in which they found that there were at least 4 categories of information which were being shown to the public and really used by teachers. And that was that there was a few things in the world that were really well established. Then there was a bunch of things that were probably so, which means that they were probably true, but there was a huge level of human variation. For example, we know that sleep is important for all learning, but how much sleep can't be prescribed, because between 4.5 and 12 hours is normal. 8 is average, but that doesn't mean that you can prescribe and tell everybody they need 8 hours of sleep to learn well. So we know that there's things that are probably so, and those are the 2 good categories. But then we end up with things that are just intelligent speculation. You know, men and women look different from the outside, so the brain should be different on the inside. Is that always true? And then we have a category of what is just myths. So based on these myths, I want to ask you a few questions and see how you stack up in your own personal knowledge about the brain and learning at this state. So I'm just going to give you a couple questions and what's your answer on this. So true or false? Human brains are as unique as human faces. Is this something that's well established? Is it probably so? Is it intelligent speculation? Is it a myth? In general, we'd have to say that this is true because they're as similar as faces are. So while most of us have 2 eyes, a nose, 2 ears, a mouth, we all have the same parts, but we don't have identical brains. And this is really interesting to look at because there's a good reason why this is true. I mean, even identical twins don't have the same brain. So even though their faces might appear the same, their brains are going to be different. And this is really based on our perception of the unique experiences that surround us. Another question. All brains are equally prepared for all tasks. Unfortunately, this is not true. You're born, on an unequal playing field. Different people are born with different potentials for different types of things. And based on their context, the experiences that they have in their environments, they're able to maximize the potential for that genetic makeup that they inherit from their parents. True or false? Past information influences how we learn something new. Totally true. The basic physiology, you learn about your world through your senses, right? And there's no way you can bypass this first check in the amygdala and then goes to the hippocampus for confirmation. Do you already know something about this? Your brain is incredibly efficient, and the very first thing it tries to do is to see, is this like anything I already know? So association is one of the best ways that the brain learns. So looking for patterns and then what is novel are the huge ways that the brain can actually advance its learning or economize learning. So yes, this is definitely true. The brain changes constantly with experience. True or false? Very, very true. And to the huge extent that if you recall in the video, there's a lot of things that will happen at a microscopic level before there will be observable behavior. So the connections have to be made in the brain before you can actually see somebody doing something new. For example, if you ever taught a child how to read, you will work at it and teach them letters and sound out words and read and read and read and read, and weeks or months might pass, and you don't think the kid's getting it. But he is slowly but surely making all of the links necessary to be able to then finally read. So we know that changes will happen at a microscopic level before they'll happen at an observable level. The number of neurons in the brain is about the same as, now here we're going to get into the neurophysiology, so let's think about the numbers here. Is it the same as the number of fish in the ocean? Is it the same as the number of stars in the Milky Way? Is it the same as the number of people in the world? Is it the same as the number of words in Spanish or the number of words in English? I think most of you have heard this sort of in the popular press, but it's very much the Milky Way, with about 100 trillion connections between the neurons. How big is your brain? It weighs about 3 pounds. It's about 2% of your body weight. It weighs about 5 pounds. It's about 3% of your body weight. It weighs about 10 pounds or 7% of your body weight, or it weighs about 15 pounds, 10% of your body weight. When I say body weight, this comes from a Science Direct quiz, and it presumes an average human being adult of around 150 pounds. So whether that's true or not, okay, so what's the answer here? Number one, it's about 3 pounds, and it's about 2% your body weight, which is really phenomenal when you think about it. Your brain controls what you do to your body, but what you do to your body in terms of nutrition, rest, sleep, exercise, influences the potential of your brain. So it's a kind of disproportionate relationship here. True or false? Brain parts work in isolation. You have this logical, totally together left side brain, mathematics, all the rest of it, and you got this free and crazy right creative brain. Very, very false. There are no brain parts that work in isolation. There's nothing that you do that is not involving actually both hemispheres. So you're always crossing back and forth. So better brain imagery is helping us get away from this false idea of localizationalism, that X part of your brain does whatever. We are noticing that there are hubs, as I mentioned before. For example, Broca and Vernix area are very important for language. So we know that a lot of the networks repass through those areas. So we know that there's certain pieces of your brain that are vital, but they have to work within a network. So we don't say now that, here's math and here's, spatial abilities or whatever is in one part of your brain or not. That is just not true. True or false? The brain is highly plastic. It's highly flexible. Absolutely. And until you die. Your brain is plastic. This is something we're going to talk about a lot more in week 3. We're going to dedicate week 3 to neuroplasticity because this is actually one of the most fascinating areas of research that exists. And we're going to highlight a couple of the case studies that come out of that, especially the work of Norman Dodge and Paul Baki-Rita, some of these real pioneers in this field who have actually shown how you can rewire your brain when parts of it might not work for some reason. For example, a person born congenitally blind can learn to see because all learning passes through the senses, and if one sense isn't working, maybe you can learn to use the other senses to compensate. So we'll discuss that in week 3. So what's the basic terminology? If you recall from the video we just saw, there was all of these words were used, but I just want to put them out and line them up together so that you can see from the really micro level of discussion up until the more macro level at a brain level. So we've talked about synapses, which we said occur in chemical and electrical ways. These synapses work together to create different pathways, and multiple pathways will then create the networks in the brain. So this is what we're looking at here. So let's pull these apart a little bit further with some other terminology. Neurogenesis means the birth of new neurons. A neuron is nothing more than a cell in the nervous system, okay? So you all know what cells are. You have cells in your skin. You have cells everywhere. A neuron is a cell in the nervous system. So neurogenesis means a new cell birth. So we didn't believe, we once thought that, you have certain number of neurons, and then if they die, and you never have them replaced. Well, that's not true. There is neurogenesis. It does occur. It's not frequent, so you shouldn't rely on it. You can't, I'm going to go out and party this weekend because I'll replace my neurons. That just doesn't happen in that scale. But it is important to recognize that neurogenesis does exist. So like other cell generation, the brain does generate new cells. Neurogenesis, this was documented in animals. It was established in mammals in 1992, and we finally documented in humans in 1998, but not all over the place. We find it in the olfactory bulb, we find it in the hippocampus, but not in many other places has it been well documented. So it's very important to recognize that, yes, neurogenesis exists, but it's not well documented. It's not as prolific in all areas of the brain as one would hope or expect. There have been some studies recently conducted that actually look at how neurogenesis can be enhanced through enriched environments or through exercise, for example. So these are some very interesting areas of new research that might be something that you would like to consider for your semester project. Synaptogenesis is different from neurogenesis, right? Neurogenesis is new neurons, new cells, and synaptogenesis are new synapses. These are the new connections between the neurons in your brain. The formation of synapses between neurons is what we call synaptogenesis. So you have electrical synapses or you have chemical synapses. For new learning to occur, and this is how plasticity occurs, is through synaptogenesis, through the creation of new connections in the brain. That is plasticity. But we realize now that there's definite connections that inhibit this. Stress and depression decrease synaptogenesis. Other drugs, for example, will increase synaptogenesis. So we know that there are risk factors that will prevent new learning in the brain, and we also know that there are protective factors, things that will help us get to neutral or in a better place to actually enhance learning. We're going to look at that throughout the rest of the semester as well. In the older studies that we had, actually dating back to the 1940s, believe it or not, 1939, 1967, Connell tried to enhance the Golgi stain preparations to actually show between newborns, 1-month-old, 3-month-old, 6-month-old, how there were different changes at the different levels, cellular levels within the brain of increases in synaptic activity. And one of the findings that was actually disturbing to many people is that we found that the older you got, then so you were losing brain cells. But this is not what it's all about, really. What you really need to look at is it's really actually good that your brain prunes and refines its activity. It's not that more is better, not having, more brain cells. What's more important is how they are connected, and so the strength between the different areas. So in studies that are coming out now, so you'll see there's a big difference. Before we were talking only about neurons and synapses. Now we're getting into this really refined discussion about the spines and spine pruning and how that actually changes with the efficiency of the adult brain. So we know that, yes, at birth, you're born with more neurons than you'll ever have in the rest of your life. There is massive cell die-off, at birth, just contact with oxygen. But after that, then, while quantity is important, it's actually more quality. How are those neurons then going to be connected in different areas, in different ways, in different places? So we'll go into this a bit further when we talk about neuroplasticity next week. So the big takeaway from this class is that it's important to recognize that not only is the proliferation of synaptogenesis or new connections important, but also pruning is really important. Pruning leads to efficiency in the brain. So we know that when different types, and there's the Hebbian synapse hypothesis in 1949, Donald Hebb had this idea that, the cells that fire together wire together. So when they're connected, when certain occurrences happen, then neighboring cells will also connect. If you remember the Pavlovian dogs, right? They ring the bell, the dog salivates because he thinks he's going to get fed. But then you ring the bell and there's no food, the dog will still salivate because he's connected two things together. So this has a lot to do with traumas, therapy, things that people might experience in their life. People walk down a dark alley and they automatically think they're going to be, they get into a fear mode because once upon a time they were jumped. So they connect certain things together, and so they've got a connection going between that experience and their reaction. And the other big idea related to Donald Hebb's work has to do with this concept of use it or lose it. Some of you might have taken a foreign language when you were really little, but you haven't used it in, a decade. So you're really hard-pressed to find it, and you think you don't have that language anymore. And it's probably true because your brain is really efficient. If it doesn't see that you're using it constantly, it will relax and either connect to something else or not connect at all. But that strengthening of the connection, which is called the myelin sheath, which we'll look at just a second, doesn't exist. So it's not easy to recall things that you haven't used. So if you want to remember something, you really have to be using it all the time. So here's some vocabulary we're going to be using a lot throughout the semester, and I hope that you dominate by now, okay? The neurons are the cells in your nervous system. The dendrites, or spines, are what come out of or connect from the neurons, from the cell body of the neurons, and they make synapses between other groups of neurons. And when the synapse is strong, you'll actually have a myelin sheath will develop, and that is actually white. This is called the white matter. The gray matter is actually what is the cell body. So when we talk about white matter and gray matter, we'll see that when you have something with more rehearsal, remember we talked about use it or lose it, the more you use something, the stronger the connection. And there's this kind of fatty tissue, it's called a myelin sheath. It actually speeds up the connections between the different groups of neurons. So when the myelin sheath has been rehearsed enough, it means that you can find something really quickly in your mind. But if you haven't practiced that foreign language for a long time, this will debilitate and there will be no quick connection. You will have to dig to find that information, okay? So we know with more rehearsal, there's more myelin sheath, which means there's greater white matter, which means there's speedier recall. And this is actually seen now in more up-to-date studies. This is just really from 2015. But the idea is that we have these models now, and we're trying to establish them in real physiological models, which is really difficult. But we have now at least these caricature models where you're saying, here's a hypothetical circuit. And before we have learning, there's some things that are not really connected. But then once we actually have learning process, then we've strengthened certain connections between different points, which will increase the speed with which we can retrieve that information. We'll talk about this in another class, but memory is fundamental to learning. So being able to retrieve something out of memory is fundamental to your ability to learn new things. Finally, lobes and cortices. In your cerebral cortex, you have 4 lobes, right? So we have the frontal lobes, which takes up the greatest amount of real estate here. The parietal lobes, which goes across the top of your head. Temporal lobes, just between your ears and back. And the occipital lobe. And we'll pull each of those and the networks related to them apart in the example we're going to give you when we talk about neural networks in just a second. Inside of the brain, you have a limbic system, and this is really key to the discussion we're going to have about affective neuroscience and emotions. We know that the amygdala is key for processing emotional memories, the hippocampus for long-term declarative memories, and the cerebellum, which is behind here, which is very much related to procedural memory. What we want you to be able to do is to recognize where these lobes are. And when we do discuss things about executive functions and use of frontal lobes, we would like you to be able to identify where the frontal lobe is. So we're not going deep into neuroanatomy, but we do want you to have these particular words in your vocabulary. So in the general sense, then you have the frontal lobes, parietal lobes, temporal lobes, occipital lobes. You have the cerebellum here. The pons, this is what goes down back into your spinal cord here. And then you have the amygdala and hippocampus, which are key to our discussions that we'll have here. You'll see mentioned in a lot of the literature that we have the cingulate gyrus, or the anterior cingulate gyrus, which is used a lot for different processes that are now coming to light recently, especially when we talk about the reading brain and the math brain. But we want you to be able to manage this particular type of vocabulary. So if you know the words on this page, you will be just fine for this class. So don't worry too much about these things. We're not going to drill it to death, but we do want to be able to have these typical conversations where we discuss the articles and you're very clear about what part of the brain we are talking about. Finally, the last point about neurophysiology just has to do with the central nervous system. The brain obviously is part of the central nervous system, the spinal cord. And then we have the peripheral nervous system, which has to do with the cranial nerves. There are 12 cranial nerves in your brain. They go up to the base of your brain, and they react to your ability to perceive and understand from the outside world based on your senses. So there will be reference to those things as well. Okay, so let's go back to neural networks. Remember we talked about the neurons, and we talked about or the spines, and we talked about the synapses that can occur. And multiple synapses then will create these types of networks. And in the networks, as we mentioned before, we can now find hubs, or where certain parts of the brain are connected with different types of activities to a greater extent. And in the Connectome project, you can actually see how this actually evolves from the individual synapses into a greater network system. So we know in a simplified version of the brain, we know that networks are responsible and they explain motivation, drive, thought, and behavior. We know that the networks help us understand ourselves, so it gives us the power to actually reflect on our own activities, our own actions. And the networks also allow us to manage our own behaviors and to manage behaviors of others. In a simplified version, there are some people who talk about 3 different types of networks. These are grand networks. These are the recognition networks, so what's going on in the world. The affective network, why is something happening? And strategic networks, how is something really working? Well, if you look at these sort of broken down, the recognition network is basically how we perceive our world and understand what's going on. The strategic networks are how we're going to react to that. And affective networks have to do with the reasoning behind those choices. So that's one way to approach neural networks in the brain. Other ways are to look at actual activities that might occur in your daily life. For example, the biggies of language and math are really being undertaken right now to a great extent. And what does this mean? in an observable way, what you and I can all do is that we can see that when a child is learning to read, for example, one of the competencies they have to dominate is that they can sound out a word based on the phonemic understanding, okay? So that would be something we could see. Well, what is the pathway? The pathway has to do with word and phoneme recognition, and this is something that has to occur as well as spelling, intonation, before the child can actually learn to read, which is under this big, huge umbrella of language, right? And this is a paper I presented in the World Educational Research Association in Hungary last September, which had to do with breaking down these neural networks into different pieces. So, for example, in the math understanding, what do we know so far about the math brain? We know that there's different areas of the brain, for example, related to arithmetic processing. And there's such a thing as a triple code, which is phenomenal. Stanislas Dehaene and colleagues found that the brain actually processes this concept of the number 3 differently if it's written in Arabic numerals, or if it's written out in letters, or if it's just in symbols. The brain is actually processing the concept of 3 differently. There are other studies that actually show mental processing. When you give somebody, 239 plus 58 equals what? When they're doing mental processing, we can see what parts of the brain, what networks are stimulated, or what pathways are occurring there. When somebody does magnitude estimation, how do they first, evaluate what they're doing, and then how do they actually quantify what's actually going on? So we know that there's certain nodes that are key there in those pathways for that type of mathematic understanding. Numerical cognition, understanding different forms of numbers, whether or not it's written as a Roman numeral, an Arabic number, or different symbolic representations. We know that multiplying numbers is very different from doing spatial tasks, like can you rotate this cube, 180 degrees, and we find similar things when we look at language networks. There's a word form area. It's in the base of the brain, near the occipital cortex, which actually helps translate, and we're understanding then what does this actual word mean. So it's a semantic understanding based on phonemic or joining of phonemic connections in the brain. And if you look at the brain as it's trying to learn a new word, how does this actually occur? So you see that there's lots of different pathways that have to be functioning. You have to actually be able to perceive, to hear, to break apart the phonemic breakdown of the word, to actually understand the lexical phonology of the word. So all those phonemes put together come together to mean what kind of a word, right? And so actually, once you've heard it, you have to actually have a semantic understanding. What does it really mean, right? So all of those pieces have to come together just to learn a single word. And if your brain is going to learn to read, an excellent book I highly recommend is Stanislas Dehaene's book on reading in the brain, where you're trying to look at all of these different pathways that have to occur for the brain to read. And that's only in their first language. Can you imagine what it is to read in a second language? Very similar networks, but overlapping, but not identical, right? So there's a lot of things that are happening in your brain when it's reading, and so it's a much more complicated task than it might first appear. Sentence comprehension involves so many different pieces of your brain working all together, as does semantic retrieval, when you're trying to remember what does that word mean, that's tip of the tongue phenomenon when you're trying to find the exact word or a synonym for something. It's actually, you have to dig deep into your brain to do that. General language processing, passive reading of active words, active reading of passive words. They do different things in the brain. So these are different parts of the brain that are used. So, and you pick it apart, you say there's also other pathways for spelling, for humor, first versus second language, prosody or intonation, hand gestures to meaning. There's tons of these studies on language in the brain, which helps us come to 2 big conclusions. One is that it's not simple. It's not that reading in your brain is in one space, right? We now know that there are multiple. There's at least 16 different neural networks with various pathways that are related to that. But the second big idea has to do with neuroconstructivism, that some of these pathways have to be in place before higher order pathways can actually occur. This is Siros's work in 2008. We have to build upon these things. So if a person does not have a clear symbol to sound understanding, they will never get to the level where they can interpret words, which means that they will never be able to read correctly, right? So there's a hierarchy in the order of these different types of learning stages that an individual has to pass through. So a final takeaway from this is that it's not, again, it's not as easy as it appears. Nobody can say, oh, well, get your, put on your thinking caps and get your reading brain going. There's so many different parts to it, okay? So the common assumption used to be that There were these bottom-up and top-down activities. So some things that just came from your gut, went up, emotionally processed, and then we had to react to them through perception things. And the other things were top-down, where we had to have higher order cognition, pay attention to things, and then react to them. Well, now we know that all of these things are actually occurring simultaneously. We have bottom-up, we have top-down, we have parallel processing in the brain at all times. What's also very interesting to see, and remember we talked about pruning a bit, going from novice to expert is fascinating. If you look at a brain just beginning to learn how to read, for example, it's all over the place, right? Because you're trying to develop these networks. But once the networks are developed, it's a very precise thing. And so an expert's brain is actually less active than a novice brain doing the same activity, because the expert has defined the clearest route, the easiest network to follow, whereas a novice brain is still experimenting and trying many different ways to get the information into their brain. So we know that if you look at novice to expert studies on the brain, you'll find that there's lesser brain activity in expert brains than in novice brains doing exact same activity. Okay, so what does all this mean for mind-brain health and education? Students learn and are motivated to learn in different ways. We know that, right? In some ways, you'll see that a bottom-up approach works best, or a top-down approach works best. But you'll have to agree that they're all going to be needed at different stages. So you can't just take one interpretation and say, well, we have to do this very cerebrally. Let's explain all the grammar to the kid and have them, work at it from that angle. And you cannot just throw the kid into a book and say, just figure out and read on your own. You have to do some kind of maneuvering on all these different levels in order to react to the different networks that need to be developed for simple processes like reading or doing math, for example. But the bottom line here is that there's different ways in which these neural networks are going to function. They react to different types of stimulus that we are giving as teachers or as entities in the environment external to the individual, and we have to combine all those things to make sure that they can learn in the best way possible. So we should appreciate the complexity of the brain. There's a lot of perception, what's going on. There's a lot of affective processing. Why is something happening? There's strategic things, how do I go about approaching these? There's different neural networks that are employed for different types of learning aspects, and all of those things are important to take into consideration simultaneously as we take on any new learning in our life. So what did we try to do here? At the very beginning, if you recall, we looked at this video and we talked a little bit about this, a couple of very important concepts and key attitudes that we hope that you maintain throughout the semester and throughout your life as you approach information about the brain and learning. The first very big idea was to be cautiously optimistic. There's a lot of great things that are happening and new research that's coming out and great technology that's advancing our understanding about the brain and learning. But we are really just beginning to uncover, all of the complexities of the brain. So the second big idea is that most things that we used to believe could be explained away very simplistically are just not true. The brain is incredibly complex. And for something as simple as reading, for example, you're going to have multiple neural networks and hundreds of neural pathways and millions of synapses involved in that single process. So one thing that I hope you take away in a very positive sense is that your brain is incredibly complex, and it's fascinating, and it's wonderful that you are interested in studying this and learning a bit more about it. And we have to appreciate, it's not easy to get all of these things under our belts and be able to manage this type of vocabulary as we try to explore this in a more complex way than we ever have in human history. So part of this is getting away from the neuromyths that still plague a lot of the literature, especially literature that's that teachers are exposed to. I think the teachers need to really celebrate the fact that they are actually dealing with probably the most complex organism in the entire universe, and they should learn more about it. And so we have to get away from the neuromyths, and some of them we talked about here today. We will do more of that in the section that we have this week. We also looked at the history of brain imaging, going from autopsies from more than 4,000 years ago to the present, with the hopes of better appreciating how far we've advanced, but really how far we still have to go before we can really understand the true complexities of the human brain. We looked at some aspects of neuroanatomy, neurophysiology. There are some key words there, terminology, that we really want you to dominate. And we looked finally at a couple of examples of neural networks, for example, that of reading and of math. So hopefully that we drive home this point about the complexity of certain activities that we presume occur in our schools all the time, every day, but how difficult they may be. Okay, we'll take questions now. And for those of you who want to, during your reflection time, if you find that you come up with other things that, and are not able to share them here live, please go ahead and write me an email on this, and we look forward to continue working with you. Thank you very much.